אז לצערי אני לא אני מהפלג החולוני, יותר דל במשאבים. Okay, everyone will start. Let's start. I'm very excited to welcome Avital Hahamy. Dr. Avital Hahamy is from right, studying real-world behaviors and associated brain activity, both in neurotypical individuals and clinical populations. Really, the breadth of her work is incredible. She has a PhD from the Weizmann Institute with Rafi Malach and did her postdoc in Tim Burns' lab in the Wellcome Center at UCL. And she currently, very recently, opened her own lab. She leads a group as a Royal Society Dorothy Hodgkin Fellow at the Institute of Cognitive Neuroscience at UCL. And we're looking forward to great talk. Thank you, Ayara.
And thank you, everybody who have joined us. Wait a minute. So I'm Avital. Can you hear me? Yes, okay. I'm Avital. I'm from UCL. And I want to tell you about how I believe that reactivation in the human brain connects the past with the present. But what I'm actually interested is in how you will make sense of my talk. So how will you make sense of my talk? This is me. I'm giving a lab meeting like during COVID sometime. And this is Alon. He was part of the group I did my postdoc in. And he's trying to make sense of what I'm saying. And although Alon perceives what I'm saying in a continuous way, much like you right now, according to the event segmentation theory, his brain is segmenting my talk into discrete events. So that's, for the sake of argument, assume that this sound wave represents my talk. And let's assume that each chunk here is not a word. Let's say it is an event. An event could be, for example, a slide. So the first event would be slide 1, my title. And then event 2 would be the intuition to the big problem. And when I'm switching slides between slide 1 and slide 2, your brains would detect an event boundary. That's the end of one topic, one event, and the beginning of a new topic. So after slide 2, the intuition to the big problem, there will be an event boundary, maybe because someone will make a joke. And then later on, there will be an event boundary, and slide 3 will present a literature review, and then event boundary, and what's still missing, and how I propose to solve the problem. Now, according to the event segmentation theory, what's going on in your brains is basically you're extracting the gist of each event, and you're storing that gist of each event, of each slide, into memory. But isolated event representations won't help you understand the narrative, right? Because if we're on slide 4, and I'm saying what's still missing, you need to know what is known. And what is known was actually presented in slide 3, the literature review. And to understand how I propose to solve the problem, you need to know what the problem is. And that was actually presented in slide 2. So how do we form these links between different events in an unfolding experience? Or how do we bind related events across time? There are several clues that led me and my co-authors on this paper in our search. The first is replay at event boundaries. So replay is heavily studied in rodents for many years now. We know that in the hippocampus we have play cells, and these play cells fire a specific receptive field that is specific to a location in space. So for example, if this rodent is running through this track, then a certain red cell would fire for this specific location in space, and this pink cell would fire for this specific location, and so on and so forth. But there are certain times when a rodent is pausing or receiving a reward, or at the end of trial. And these are very similar to event boundaries, the ending of one event, the beginning of a new one, that we see the same cells firing in the same order, in a compressed way. And this is what we call replay. Now, this replay is well studied in rodents, but there is also a similar phenomenon that is studied in humans called reactivation. We call it reactivation because there is no serial element here. We don't have access to the actual serial firing patterns of neurons, but we can find correlations. So there is this one paper that showed that while people are watching a movie, then at the endings of scenes, at event boundaries, we see the reactivation of the just ended event. This means that whenever your brain detects an event boundary, there will be this cut, and the just ended event would be replayed during this event boundary. And in both scenarios, both in rodents and in humans, it is believed that this replay has an offline function. It chunks events and stores them into memory. And this replay could, in the rodent literature, is believed to build an internal model that allows you to plan ahead or to make decisions, but it's an offline function. So just to help you understand what I mean right now, I have just switched slide. That means that your brain should detect an event boundary, and you should have a replay of the just ended event, of the slide that you have just listened to. So you should replay the fact that there is replay in rodents during contextual shifts in ongoing experiences, and there is reactivation in humans during contextual shifts in an ongoing experience, and both phenomena are presumed to have an offline brain. So this was clue number 1, replay and event boundaries. Clue 2 is that both event segmentation and replay engage the default mode network. So this is the default mode network. It is a set of brain areas that, at least in fMRI, was proposed to be an offline network. And the reason is that when using fMRI, we compare brain activity usually to a resting baseline. And some areas during task increase their activity. But these set of areas actually decrease their activity in certain tasks in fMRI. This is why it was proposed to be an offline network. But we actually know that there are certain tasks that engage this system, that causes these areas to increase their activity. One of them is, while listening to a narrative, is event boundaries. So we know that when people listen to a story or watch movies, during those event boundaries, these brain areas actually increase their activity. So we know the default, and I should say that the most pronounced areas that we know that increase their activity during event boundaries are this angular gyrus and precuneus or retrospinal cortex and hippocampus. Now we also know that these brain areas are somehow related to replay. And we know that both from rodent studies and monkey studies and human studies, we know that when we detect sharp wave ripples, which is when we tend to find replay events, then some brain areas increase their activity, and those that mostly increase their activity are areas of the default mode network. This is work done in mice, and you can see the retrospinal cortex here that shows the most increase in activity during sharp wave ripples. So the default mode network seems to be related both to event segmentation and to replay. And I remind you, the previous clue was that replay or reactivations occur at event boundaries. The third clue is that offline replay binds related events. And this is a beautiful study done in rats, where rats are given this maze. It has two compartments, and they can either go through this compartment of the maze or through this compartment. What they never, ever, ever do is this. They can go through the outer bounds of this maze. And then the rats sleep, and during sleep, we can find replay events that represent this compartment or this compartment. But the interesting thing is that we also find replay events that represent the outer bounds of the maze, the one that was never experienced. And the authors here actually propose that perhaps these replay events can piece together different parts of our experience. In this case, it's the spatial experience, right? Like constructing a mental representation of space. So if you want to go from one place to another and you can think of a shortcut, you can think about it without ever taking that shortcut. This is quite similar to how they interpret these data. But navigation is also an unfolding experience. So perhaps the same machinery can be used not only for spatial navigation. Perhaps we can piece together different experiences that unfold in time, like narratives, to create a mental representation of that narrative. Now, here we are faced with a technical challenge. It's quite difficult to move from the rodent, very accurate and precise measure of neural activity to the scale I'm working on, this meso scale of fMRI. So I'm going to present a method that me and my previous supervisor developed that may probe these replay or reactivation events. So offline replay binds related events. Can online reactivation bind related events? And I remind you that right now you're not experiencing offline replay events, right? At those event boundaries, when I'm switching between slides, this is an online experience. You are engaged, hopefully, in my talk. So when I'm telling you that, I'm telling you what's missing and how I propose to solve the problem, you should be creating these links to the relevant parts of my talk. So the question is temporally remote information reactivated at event boundaries of naturalistic experiences like this talk? And again, to clarify what I mean, this is an event boundary. I've switched slide, and if my hypothesis is correct, that means that right now your brain would be reactivating not only that just ended event, the previous slide, but relevant pieces of information throughout my talk. That the human brain parcels ongoing experiences into events, and there is online reactivation of the current event at event boundaries. But to make sense of ongoing events, temporally remote information must also be replayed. Rodent replay can link remote spatial events. Can human reactivation link remote episodic events? So our experimental approach. We have chosen to analyze narratives, and why? So in all these replay experiments done in rodents, the main paradigm is usually to use repeated events. So a rodent would go through a maze again and again and again. And that would mean that repeated experiences would also have repeated representations in the brain. And it would be hard to differentiate between different episodes or different events. But in unfolding experiences, like narratives, each event has a separate representation that is unique to each event. So potentially, we would be able to detect the integration of different representations of events across time. We have used two data sets made available to us. One is of people listening to a story, and the second one is of people watching a movie. And these two data sets are quite different from one another. They're different in the modality. So here, this is only an auditory stimulus. This is both auditory and visual. They're different in their content, they're different in the structure of the narrative. So this is a linear narrative where one event leads to another. And here in the story, we have two unrelated storylines, and they're switching. You hear one paragraph from one story, another paragraph from another story, and they change until they integrate the very last section of the story. These data sets are different in the structure of the stimulus. The events here are quite controlled. They're quite similar in their length. Here, it's more random. The pre-processing is different because this one came pre-processed and I pre-processed this one. And the reason I'm boring you with all these technical details is just to say that these data sets are so different that any consistent effect across the data sets will tell us something general about neural processing of naturalistic stimuli. So the methodology is representational similarity analysis. I'm going to give you a brief introduction to that, just because I don't know if everyone is on board. Let's say that this is my movie. This is one scene, this is the other. And I'm marking an event boundary in the transition between scenes. It's an instantaneous transition, right? It's not as if one scene ends and then there is a blank and then you continue. No, it's instantaneous. And in fMRI, we have a relatively low temporal resolution. Our units of measurements are called TRs. In these two data sets, each TR is 1.5 seconds. So these event boundaries, we're going to find the right TRs that contain them. And then we're going to take this TR and shift it by 4 additional TRs. And the reason is that the bold signal is very slow. So if you present a stimulus in a scanner, let's say a face, it will take around 6 seconds until you would find the peak of the bold signal, which is why we're going to shift the timing of the event boundary by 4 TRs. And now we're going to run a whole brain analysis. We're going to take each voxel, that's a 3-dimensional pixel, each voxel in the brain, and we're going to create a sphere around it. And for each point in time in the fMRI data, all these voxels are going to have different levels of activity. This is what we call a representation. So let's say this is the representation of this sphere in the brain during this event boundary. And now I have the entire narrative. EB here would be event boundary. So for each event boundary, I will have a representation, and I'm spreading it into a vector now. So it basically tells you how active each voxel in a sphere is. And I'm not only interested in the representations of the event boundaries, I'm also interested in the representations of the actual events. These are the scenes. So to get these representations, I will average all of the time points, all of the TRs within a certain scene into a single representation. And that would mean I will lose all the fine-grained temporal information within these scenes, but I will be left with something more general. That could be the gist or the context. And this is something that is very commonly done in the naturalistic imaging literature. Okay, so this is about representations. Let's talk about the similarity analysis. So I have my representations of event boundaries and of scenes. Now, reactivation would entail that during an event boundary, I will find the representations of previous events. And that would mean that this event boundary, for example, would be correlated with previous scene representations. So what I've done next is to create this similarity matrix. It's just a correlation matrix between event boundary representations and scene representations. And let's think about this matrix for a second. It has 2 parts. It has the lower triangular part, which is the past part of the matrix, because if this is an event boundary, this is the correlation of each event of this event boundary on all past scenes in the movie. And this upper triangular part would be the future part of the matrix, because this event boundary will show me the correlation with each future scene in the movie. So past part and future part. I'm also removing the main diagonal that represents the correlation between each event boundary and the just ended scene. And the reason is that I'm not interested in the just-ended scene. I want to ask if remote information is reactivated, not only the just-ended information. So let's assume this is my talk. I've given you an introduction to event boundaries, and then I told you, well, maybe there's replay event boundaries, and maybe there's replay of remote information during those event boundaries. If my hypothesis is correct, when I'm telling you that there is replay event boundaries, you would need to understand what event boundaries are. So you would need to reactivate this part, where this scene that I gave you the introduction to event boundaries. And when I'm telling you that there's replay of remote information, you need to know what replay is, what event boundaries are, so you would specifically reactivate those parts of my talk. This is an example of such a matrix from actual brain data, and you can see there's quite a lot of variability in the entries in this matrix. So indeed, some scenes are reactivated more or correlated more than other scenes. But there is a problem here, because these correlations could simply reflect scene similarities. Here's the thing. The fMRI signal is highly autocorrelated, and it has very low temporal resolution. So this event boundary is actually quite correlated, so the representation of this event boundary is quite correlated with the representation of this scene. And this scene representation could be correlated with previous scene representations because they may be similar. So I'm telling you, well, yes, I'm going to correlate event boundary representations with scene representations. In fact, you would be correct to say, well, actually, you could merely be correlating scene representations between themselves. But if this is true, then this similarity matrix would be reduced to a scene-by-scene correlation matrix, right? And a scene-by-scene correlation matrix is symmetrical because the correlation between scene 5 and scene 2 is identical to the correlation between scene 2 and scene 5. So what I'm going to do to control for this possible confound, I'm going to take the upper and lower triangular parts of the matrix, and I'm going to subtract between them. That means that any symmetries in the matrix will be lost. Any scene similarity effects will be accounted for. I'm controlling for them. So as a first step, I'm going to ask whether on average I will find reactivation of past events. So I will average this lower triangular part of the matrix and this upper triangular part of the matrix, and I will subtract the two to get a single number. This is very similar to actually subtracting the upper from the lower triangular part. But this will give me this one number, this average effect of whether I find reactivation of past events, which I can then assign to the voxel that started my analysis. Remember, I started with a single voxel that had a sphere around it. This is all done for a single voxel. I'm getting a number, I'm assigning it to my voxel, and I'm giving it a color to represent the value of this index, where warm colors represent reactivation of past events, and cold colors represent the reactivation of future events. So it's a two-sided hypothesis. And then I'm going to do that for the next voxel, and the next voxel, and the next voxel until I get a map. So I'm going to show you maps now that this is what they represent. Will I find areas that show reactivation of past events more than future events? I should also say there are some issues with autocorrelations here that I'm not getting into, but if you're interested in those, I'm happy to elaborate later on. Okay, so which brain areas do I find? This is the movie data set. And I'm not getting any areas that show me future reactivations, but I am getting these yellow areas that show me reactivation of past events. And these are the angular gyrus and precuneus and hippocampus and some other brain areas. Now I'm looking at the second data set. And again, I'm seeing angular gyrus and precuneus and hippocampus. And because I want to be very careful here, I'm only going to interpret those areas that are consistent across data sets. So I'm not going to talk to you about the visual or auditory or language areas that I found in the first data set, although they're interesting. Yes?
Consistent across subject as well?
Sorry?
Are they consistent across subject? Or is that
Yes. So this is a group map. This means that I'm running this analysis for all my single subjects, and then I'm running a statistical test across subjects to find those areas that are consistent across subjects. I can also run a mega-analysis, actually just pulling together all of my participants just to see how robust this effect is, and I'm finding the same brain areas. So it seems that perhaps these default mode areas, the same areas that show this sensitivity to event boundaries, are also regions that show reactivations of temporally remote past scenes at event boundaries. And I'm reminding you how different these data sets are. So when we first looked at this, we were quite amazed to find this replication. The next question we wanted to ask is if this reactivation is shared across brains. And why is this interesting? So far, I showed you an analysis that is done within the brain of a certain participant. So I'm taking my event boundary representations and my scene representations from the same person, and I'm calculating my reactivation index. But previous work have actually shown that scene representations in the default mode areas are shared between brains. What does that mean? That means that when you are all listening to my talk, the representation of this slide will be actually correlated between your brains. There is a shared component of these representations. So we wondered whether the reactivation is also shared. So to understand this, we ran a between participant analysis. And that means that I'm taking my scene representations from one participant, but I'm now averaging event boundary representations across the remaining participants in the same experimental group. And then I'm doing the same thing. I'm calculating my reactivation index. But the reason this could be interesting is that if I find reactivation that is consistent across brains, it is shared across brain, it could hint towards shared binding. So if during an event boundary you are uploading from memory the relevant bits of past information that are relevant for your current understanding, and if you're all uploading the same bits of information, we would have shared event boundary representations and shared reactivation. So if you're choosing the right bits to reactivate, this may be the basis for shared understanding of a narrative. So again, I have my reactivation matrix. I'm subtracting the future from the past. I'm assigning each value from each voxel to a map. This is the previous analysis I showed you, the within participant analysis, and this is the between participant analysis. And you can see these maps are quite similar. Again, we find the angular gyrus and the precuneus and retrospinal cortex and the hippocampus. This is true for the movie data set, for the story data set, and for the meta-analysis. So there seems to be a shared component of reactivation between brains. Now, is this reactivation specific to event boundaries? So I'm talking to you about event boundaries, and I'm assuming everything should be happening during that event boundaries, but maybe event boundaries are nothing special. Maybe I would find reactivation in any other point in time. So to test this, this is again the schematic of this narrative with all the representation. And this is my reactivation index as calculated for event boundaries. What I'm going to do now, I'm going to take the three areas that replicate across data sets, the precuneus, angular gyrus, and hippocampus. And what I'm going to do, I'm going to plot the level of reactivation as a function of leg from my event boundary. So my event boundary is going to be the 0 point. I'm not going to shift my signal to account for the hemodynamical delay. So this will be my event boundary. There will be some point that is basically this reactivation index, which will be plotted here. And this is the bold signal. And I remind you that for the bold signal, when you present a stimulus in your 0 time point, it will take around 6 seconds until you would find the peak of activity around here. Now, this is not just the raw bold signal. This is going to be the reactivation signal, but it is built on bold dynamics. So if there is something special about event boundaries, I would expect to find the peak of reactivation around 6 seconds after the event boundary has appeared. So how will I calculate this? Just like I calculated my reactivation index for event boundaries, I can do the same for any other point. Let's say point that is event boundary -1. So here I will have the representations of event boundary -1 correlated with scenes. I will have the reactivation index, and I will plot it somewhere here. And I can do that for any previous time point, for any future time point, so that I can get a signal, again, representing the level of reactivation. It is not the gold signal. So what do I find? This is the movie data set, precuneus, hippocampus, angular gyrus, for the within participant analysis. And you can see the event boundary here, and you can see the reactivation signal rising and peaking at just around 40 hours, which is 6 seconds. This is true, though not significantly, not significant for hippocampus, and also significant for angular gyrus. For the story data set, we have significant results across regions, and for the between participant analysis, we have significant results across all regions of interest.
Clarification, when you say significant, what is significant?
So this is a different way. So, okay, a second. I have this depth for you. I said that I'm comparing between different data sets, but I haven't proven that the same brain areas are actually consistent across data sets. So this is my ROI analysis. This is a cross-validation analysis where if I'm looking at the movie data set, I'm actually defining my regions from the story data set and vice versa. So all the regions I'm testing come from a different independent data set, and then I'm measuring the signal in the test data set. The way that I'm evaluating significance is different than how I did it for the maps. For the maps, I just looked at the single TR that would represent the 6 seconds after the event boundary. Here I'm doing something different. I'm taking 8 TRs after the event boundaries and normalizing by the pre-event boundary time. So I'm getting an estimate of how, of something like, of the level of positivity of my signal after the event boundary, and I'm doing that across participants. Yes?
Is this averaged over all events, or?
So yes, so this is an event boundary. This is like a trigger average analysis in classic fMRI. I'm taking all event boundaries, and I'm taking all signals, and I'm averaging them. Yes.
For the within participant analysis, how can you explain that the magnitude of the is by 2, or between the sound and the video is
Between
2
the?
orders? Between the story
Yeah.
and the video, it looks like 2 orders of magnitude.
So these data sets are very, so I should say something about the story data set. The story data set has some inherent confounds to it that, based on simulations, actually work against us. So we would expect to find reduced effect for any analysis in this story data set, mainly for reasons of autocorrelation. I'm happy to explain it later. But basically, it was constructed in such a way, this experimental stimulus. First of all, this transition between different stories inherently mean that if your event boundary I should explain. Okay. If there is an autocorrelation effect, it would be symmetrical between past and future. The problem is there is inherent asymmetries in this data set. An event boundary that comes right after a certain scene or a certain paragraph is actually attributed to a certain storyline. So the event boundary is closer in time to the previous, to the just ended narrative than it is to the previous event of the different narrative. On top of that, they've introduced some blank period between events, meaning that event boundaries are closer to the past than they are to the future. Hence, this data set shows us reduced effects overall. Yeah. Yes.
If you notice, like in the one in the middle, there will be 5 times more volt.
So I can't hear you, sorry.
The number between 0 to 5 in the region.
This one?
Yeah, 5%, 5 times more signal.
No, the 10 to the minus.
It's 10 to the minus 3.
Okay.
So this, remember that.
Okay, it's very high.
So remember that this reactivation index basically means I'm subtracting correlations from one another. So the magnitude of difference wouldn't be very high. It's not like a bold signal.
And if you try to move the event, see if you find like something like that anyway.
What do you mean move the event?
You put here the boundary and it looks, well, differently after it, but the other option is just do some kind of reshuffle of when this event.
Yes, so when I shuffle events, if I create synthetic event boundaries, then I don't get funds. Yes. Any other questions? Okay, so the last question we asked in this paper is whether reactivation is content specific. So if we're in slide 4, you definitely want to reactivate what's known to understand what's missing, and you definitely want to reactivate what's the problem to understand how I propose to solve it. What you don't want to reactivate is the joke. Some information is relevant, some information is not. And for this to be considered a meaningful mechanism, we would want to find selectivity in the content that is being reactivated. So is the reactivation content specific? How do we approach this? So we have the description of the movie. It's not just the dialogues, it's actually what's happening on the screen in each point in time. So something like, we can see Sherlock walking in, he sees Watson, he's sitting on a chair, and then Sherlock says blah, 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 blah. So we have the text representing the movie. And for the story, well, we have the text representing the story. And we have these, our lives have changed in this NLP revolution, so we have so many ways of going about this, but I'm going to count words. So I'm happy to tell you why I count words later on. There are different justifications for this. The first being that I've done this before ChatGPT entered our world. So the approach that we use is called a bag of word approach. What this means basically is we're going to take all the words that appear in a certain narrative, we're going to put them in a bag. And we're going to create this big, long vector representing each word. And then for each scene in the movie, we're going to count how many times for each scene has this word appeared, and so on and so forth. We're going to normalize this, create probabilities, and we're going to take these bag of word representations of scenes, and we can correlate them to create a similarity matrix between scenes. And this is a nice sanity check. So this is the story data set. Remember, it has 2 storylines and they're alternating. And what you would expect to find is some kind of a checkerboard pattern, right? Because scene 3 should be correlated with scene 1 because they're the same narrative, but it shouldn't be correlated with scene 2 because it belongs to a different narrative. And a different narrative means different characters, different locations, and different topics. So seeing this checkerboard pattern is actually quite encouraging. So we're going to take this scene similarity, this content similarity matrix, and we're going to take our brain reactivation matrix. We're going to correlate the lower triangular part with the lower triangular part, the upper triangular part with the upper triangular part, and subtract the 2 again to account for scene similarity effects and autocorrelation effects. So again, past minus future assigned to the voxel that starts each analysis. So is reactivation content specific? This is the movie data set. This is the story data set. This is the movie data set, this is the story data set, and this is the mega analysis. We have the within participant analysis and the between participant analysis. We can find some interesting areas in the movie data set. Again, reduced effects for the story data set. For the within participant analysis, this does not survive correction for multiple comparisons, but it does for the between participant analysis. So it seems that some similar areas do come up as showing specific reactivation, which is nice. But the most important question is whether the same area, this precuneous area, that showed on average reactivation of past content, does it also show the specific reactivation of content? And it does, across data sets and analysis method.
What about the other 2 areas?
Sorry?
The hippocampus?
So no, so because we didn't even see them in the whole brain analysis, we didn't quite cover. So final event boundary here. I've suggested that reactivation of remote past events in regions of the default exist in regions of the default mode network. And I showed this within brains and between brains. The between brain analysis suggests that there may be shared binding of past with present information. I propose that reactivation is specific to event boundaries, as you can see in these reactivation signals. I've proposed that reactivation in the precuneus is specific for relevant semantic content based on a bed of word analysis, which means that there could be relevant binding of past with present information. And these results, I think, are quite robust. They're replicated across very different data sets. And our interpretation is this: well, reactivations have been suggested to have an offline function, right? But we find they operate on the fly while you are understanding the narrative. So perhaps reactivations could be an online mechanism that binds information across time to interpret the present experience in light of past event and then consolidate events within their context into memory. But there's also something that I think is quite interesting here, and that is, if you think about these specific points in time, these event boundaries that are correlated with relevant past events, these could be, in a way, thought of as a compressed version of a narrative, right? So if you think about a model, like a cognitive model, like a spatial model, that model would basically contain the transitions between possible locations in space. And these event boundaries potentially could represent the transition between relevant past events. So I'm going to show you some very initial results now. Just to remind you, I've opened my lab 2 months ago. So please take everything from now on with a grain of salt. Well, take everything with a grain of salt, but even more so with the 3 more slides that I'm going to present to you. So very initial stuff. So we are wondering whether event boundaries represent a compressed model of evolving experience. So all these theories of cognitive maps and the way we study them in rodents is quite different from what I did here, right? Usually we have a rodent and it has some experience with space, and then we measure these replay events while the rodent has already acquired a model. But this is quite different than watching a movie, because when you are watching a movie, it's the first encounter you have with the experience. So presumably you would be building your model here, but it's not the same thing that we study in rodents. But the cool thing is that this data set of the movie, it actually has a second part that I haven't analyzed yet. The second part is, so after participants watch the movie, they go into the scanner again and they're asked to freely recall the movie in how many details they want. So it's quite interesting. Some people take as much time as it took them to watch the movie to describe it. And some people give a very concise version of the movie. But in a sense, we have access to their model, which they have acquired while they watch the movie. So during the movie, I could parcel my signal into event boundaries and scenes, but I can do the same for the recall signal too. This time, the event boundary is not determined by some external cue, which is the transition between scenes. It's something you determine. When you stop describing one scene and then start describing another one, that would be an event boundary, but it is internally generated. And this data set has been used before to study the encoding of scene information, basically measuring the similarity between scene representations of movie and of recall. But I'm interested in something else. I'm interested in these event boundaries, these specific points, and I'm wondering whether they could represent a compressed model of the narrative. So how do I go about this? This was done in collaboration with a postdoc called Matthias. We basically wondered, if we take our reactivation analysis, and this time we take our event boundaries from the recall data, would we find reactivation of movie event boundaries. This basically means that I'm creating my matrix, but this time I have recall event boundary representations versus movie event boundary representations. And I can do the same trick. I can subtract the future from the past and ask, would I find areas that show me reactivation of information of event boundaries? And this is what we find. So this map is blue, right? The previous map was yellow, meaning that we found reactivation of past events. But this time, we find reactivation of future event boundaries. As if when you are telling me about the movie, you are actually reactivating future event boundaries from the movie, as if using that model to navigate your story, right? So this is, of course, an interpretation, but in a sense, you need to decide how you're going to tell me the story. You know how it ends this time, so you're reactivating future information from the movie. And again, we wanted to ask if these are the same areas that show reactivation during the movie. So we used the independent regions from the story dataset. So it's not the same dataset, it's completely independent. This is angular gyrus, precuneus, and hippocampus. And angular gyrus and precuneus do show this future reactivation of event boundaries, but not hippocampus. Or we do find hippocampus, but it's not the same hippocampus. Yes?
Can you determine the timing of recall event boundaries by transition between scenes in the movie?
So we have the timing of event boundaries from the movie as the transitions between scenes. And the timing, the timing of recall event boundaries is when you told me about a scene and then you continue to another scene. So I know the scene structure from the movie and I can match it to your description of the movie. This is not something that we have done. The authors that acquired this data have done it for us. Now, it's a kind of a weird analysis, right? I'm correlating event boundaries with event boundaries, so we wondered whether this could be confounded by scene representations. So then we did the same for scenes. We took event boundary representations from the recall dataset and correlated them with the movie scene representation. So again, I have my matrix. These are representations of recall event boundaries and representations of movie scenes, and I'm doing this trick to find my reactivation index. And this time, I get a completely different effect. As if while I'm internally experiencing the movie, I'm reactivating past movie events while telling the story. But I'm also planning, perhaps using my future event boundary representations from the movie. As for the ROI analysis, here it seems to be the same areas as we found from the experience dataset. I won't call it the movie dataset because it's the story dataset. So just to conclude this part, and again, we're still thinking about it, okay? Nothing here is, like, there are wiggly lines here for a purpose. It could be that when you are using your model, when you are recalling an evolving experience, we would find reactivation of episodic events to know where you are, just like a rodent in space. It stops, it says, where am I? Let's reactivate where I've been. You should do the same thing when you're experiencing a narrative, but you could have the reactivation of boundaries, perhaps models, to plan where you're gonna go. Do I have time for a different thing, or should I stop here?
Hold on, it's 12 minutes to start.
Do you want to hear about an NLP analysis, or should I just stop here? Okay, let's stop with questions, and then if we don't have enough questions, I can. You know what? Actually, no, let's do it very, I really want your inputs on this. Very quickly. This is Ellie. She's a very talented postdoc. She is an NLP person. That's her thing. And she said, well, actually, it's pretty cool that you have the movie text and you have the recall text. We can actually compare the two texts compared to it, like to see how accurate the report is compared to the actual experience. Let's do that. She did that. This is called G-Eval. It's basically a method that uses GPT to score the accuracy of text summarization. You take a summary of the text, you take the original text, and you get a score telling you how accurate that is. And we can do that for each scene, and we can measure the correlation between scene representation and ask, would it be the case that the more similar the scene representations are, the more accurate accurate you are in your report, in the way you remember the thing. And these are the brain areas that show us this effect. Again, precuneus, frontal cortex, hippocampus, and angular gyrus. Where are you? Here you are. This does not survive correction for multiple comparisons, but it does show us these effects for an ROI analysis. Again, with a grain of salt.
And
That's it.
No negative?
Sorry?
Nothing negative?
Nothing negative. Nothing negative. And that's it. I want to thank Tim Behrens, my postdoc supervisor, Chaim Dubosarski, who worked with me on the paper that we published, Matthias, Ellie, and Chris, who are working with me now, my funders, and as I said, I've just started my lab. If any of you are looking for a PhD or postdoc abroad, I would love to speak Hebrew in my lab. Privilege. Thank you.
Yeah, very nice talk, very cool findings. Just a technical question. So in the voxel correlation matrix, why would we not expect just temporal effects? Why don't we see like similarity in close? Yeah.
You're absolutely right, and this is a very serious confound we were worried about. So should I? Maybe I can. Hang on. Let's talk about this confound. Here we go. Is there? Okay, so here's the problem. This is a problem we have when we are running this similarity analysis within brains. This is a simulated bold signal, and it has this 1/f structure, right? The autocorrelation structure would look something like this. So adjacent points in time would show high correlations to each other. Now, the first thing we do during pre-processing is running a linear high-pass filter, meaning that we're getting rid of this linear trend, but we also get this correlation structure, which is very bad. The reason that it's bad, because what my analysis is doing is taking a single TR and it's correlating it with previous events in time. And there is a temporal structure to these correlations.
So what is the structure? I see a peak at the origin.
So this is the autocorrelation structure of a simulated 1/f signal. So you can see that before applying this linear
No,
filter.
but I'm looking here at this plot.
This one? This one?
The bottom one.
Yes, this is the autocorrelation structure. You can see that
There
each
is
point is...
central peak, I understand.
Yeah, and then it has this alternating positive...
Is it significant?
Even if it's not significant, I wouldn't want it to contaminate my analysis. This is my problem. Sorry?
Why contamination? Just noise.
But it's structured noise. This is my... I don't want any structure... Because if this is what happens to a signal...
It's not structured if it is noise. If it's
But-
not significant, it's not structured. You just finite samples. You always have fluctuation, that's all.
We were still worried, so we really wanted to control for this. So the thing is, sorry? So my analysis, again, is correlating one time point with previous and future time points, but correlations between event boundaries and scenes should have a temporal structure that is symmetric in time, right? Everything that happens, if this is an event boundary, it will be symmetrical in the future and in the past. So in a sense, you would expect that if I'm subtracting the future from the past, this will be lost because this should be symmetrical, right? But here's the thing. I simulated 1/f signals that shouldn't show any reactivations at all because they're random. And I said, well, I'm subtracting the future from the past, I should get 0 reactivations, right? I should have a distribution of reactivation values that is centered on 0. But this is not what I find. So this is the movie data set. So I'm taking a signal and I'm just chunking it according to the actual timing of event boundaries. And this is the story data set. And again, I'm getting distributions that are biased. And you can see these ugly correlations across the diagonals. So something doesn't cancel out. This is a problem. The reason is that there are asymmetries in my experimental stimulus. The first asymmetry is the fact that I'm removing my main diagonal. My main diagonal is the correlation between an event boundary and the just ended event. But the just ended event is part of the past. And that means that there is more temporal distance between my event boundary and past events compared to future events. This is not good. The second problem is that there are problems with the stimulus structure, which we can't control. The first problem is that in the movie data set, the first 15 scenes are very short and very unrelated to the rest of the movie. And that means that on average, an event boundary would be closer in time to the past than it is to the future. So again, this introduces a bias. In this data set, as I told you, they introduced a blank period after the event boundary. So again, each event boundary is closer to the past than it is to the future. So this noise would not cancel out between the past when I'm subtracting the future from the past. For this reason, we measured the extent of the noise and just removed all the diagonals that are impacted, and our distributions were then centered on 0. Now, you could say, well, maybe it's not good enough. I hear you. The good thing is that we have 2 analysis methods. The first is within brain, where all these correlations appear. The second is between brains, where there shouldn't be autocorrelations because these are independent signals, and we find replications of our effect between these measures. So we were kind of okay with, even if this is not a perfect solution. So we were happy with this. I don't even remember if that answers the question. Who asked it? Yeah, does it? Okay, sorry. I should also say about your question. So that narrative that has an intertwined, like story A, story B, story A, story B, we do find that we find more reactivation of the relevant pieces of the narrative, although they're further away from the irrelevant. So I think there is something there that is more than a temporal bias. Yes. Yes.
I'm curious to hear what you think about the fact that you didn't find any expectational signals. Like I would expect, especially in a movie, you can in these event boundaries have the expectations about the next scene. You're going to the bank, you want to see the bank. So why do you think you didn't find it?
So that's interesting. I think there are 2 reasons. So I don't know. The reason is I don't know. I mean, when I did this analysis, it was a two-way analysis. It could have been. We could have found future reactivation. I think first, it doesn't necessarily needs to be consistent across participants. So even if there are individual expectations, maybe they're not robust enough to be consistent across people, and then this is why we don't see it. The second thing is that maybe these predictions are more relevant to the short term than to the long term. And then, at least for the within-brain analysis, we're actually removing all the short-term effects. So removing diagonals means that we're ignoring the short-term.
I was actually wondering if it couldn't be biased by the actual, because expectations are more abstract. And you seem to find the correlations better when they've already seen it or heard it.
I'll tell you a secret. This is not recorded. There was this one area that showed a negative effect in the movie data. It didn't survive correction for multiple comparisons, but it did show consistency across the 2 data sets. That area was entorhinal cortex. So if you think that entorhinal cortex is basically it's coding for structure, and you have a structure in any narrative, even if you think about beginning, middle, and end, there is a structure to it, then it would make perfect sense that entorhinal cortex would show this prediction. As I said, it didn't survive correction for multiple comparisons, so I shouldn't be interpreting it. But I did, but never mind. Yes.
So I have two questions. One is more, I think, theoretical, and then you started with replay, but then you switched to reactivation, and it's a huge difference between the two. So the replay has an episodic nature, right?
Yes.
It has space and time.
Yes.
And now this reactivation is much closer to semantic, right?
I have zero evidence.
You call it gist and so on, so it's really putting them together is maybe a teaser, but not really.
It is true that I have zero evidence that this is replay. It's all circumstantial. I will say that there is a new paper who used intracranial recordings in people watching this Sherlock movie. And they found that during event boundaries, there is a significant increase in shockwave rituals. So that aligns. It's not proof, it's circumstantial, it's there. One of the things that we're going to do in my lab is to try to get to find whether this is replayed. So we have a MEG data set that has the temporal resolution to find replay, and we have the method to use it. So I don't know if there are methods to detect replay using MEG. So I have a student who's going to be doing that. And maybe a year from now, I can tell you whether I think it's really replay or not. But yes, this is reactivation. I can't really call it replay.
So the other question, which is more it's technical, but it's like about the reactivation. So I'm not sure what is the actual evidence. Maybe I missed it. The fact that it's not activation, but reactivation.
Yeah.
So where do we see that it actually died out and was actually reactivated?
Yes. So you're saying something. There could be a trace that is there. It's not.
Say you have it in different regions. You have a decaying trace that is decaying at different paces.
Yes.
So in some regions, it hasn't decayed by this boundary,
yes.
and others it had decayed. But it's nothing about the reactivation. Just you're sampling the activation at
yes.
different times.
So I agree. I think the only evidence I could present to support my reactivation claim is that in that data set that has these alternating narratives, you don't find correlations to the nearer in time scenes, but you do find reactivations of further away events that are relevant to the narrative. So in a sense, so your prediction would entail that I would find this decreasing correlations across time. This is not the case, at least in this data set with the two alternating narratives. Yes?
Just wondering how we control for
Sorry, I can't hear
how
you.
can we control or maybe we did control for during boundaries? Do people move more, or they breathe more, or I don't know, do something?
So that's a great question. So they don't move more. It could be the case that physiological signals are different. I have another student this year who's going to be running a behavioral study of people watching a movie, and we're specifically going to ask whether during event boundary we see increase in heart rates or breathing or anything of that could be.
Yeah. A little bit shift
Yeah, so in these data, the way I controlled for physiological signals is by regressing out nuisance variables such as signals from the ventricles and white matter. It's not perfect.
But are they correlated to them?
Sorry?
So you can check if this time series has some.
They don't. But it doesn't mean that there is nothing physiologically different at event boundaries. I think it could be interesting, by the way. It could be that this is the body's way of predicting a change in context. I recently read a newspaper article that claims that people with asthma have poor episodic memory, and I was like, so could it be that during event boundaries they breathe a bit differently and hence like something happened? I don't know. Could be interesting. I think this is a very interesting question.
Okay, one last question.
About the recall of the movie, I would assume that by nature, when telling a story, it would be a little bit less linear than how the movie is. For example, I could be telling about the 15th scene and mentioning in the background something that happened in the previous scene, or be reminded, oh, this happened before, it's important for now.
Yeah.
Do you address that in the boundaries? Is it a new boundary if I'm reminding them?
So there are very few instances like that. Some participants start describing a scene which make them remember that they forgot something about a previous scene. If they do that, then I take the original scene and the new recall scene and I average their representations. But you're absolutely right that I could try to see whether the linear structure of the narrative, so in a sense it's the integrity of your model, in a sense. It's the way whether you remember and the way you remember it. I think that this is something we should definitely look into.
Thank you very much.
יש קורלציות, יש קורלציות, אין זאת שאני חושבת כלומר, תמיד יש קורלציה אז זה תמיד קונטרסט, אני שואלת האם יש לי יותר קורלציה בעבר מהזמן של
זה לא תשובה לשאלה.
בגלל שזה עשיתי את זה בהתחלה, חשבתי שזה יהיה יותר טוב וגיליתי שאין שם אפקט, אין שם אפקט סיגניפיקנטי, הסיבה שזה יותר מאוד טוב, אבל איבנט באונדרי הוא במיוחד כי הוא באמצע, זה השאלה יש לי בעיה מאוד, אני יודעת שלא שכנעתי אותך שיש לי בעיה כשאני כזה אבל אני באמת מאמינה שזה לא סיפור. אם אני מסתכלת על נקודה שהיא לפני האיבנט, ההגדרה היא בקורלציה יותר לסצנות עבר מאשר האיבנט באונדרי, ופחות בקורלציה, רק בגלל האוטו-קורלציה, לסצנות עתיד.
אז השאלה אם זה Reactivation או לא,
פייר, אני חושבת שאנחנו נצטרך
למה לעשות מזה Reactivation?
אני חושבת שחובת ההפכה אלינו, אני חושבת שהדאטה של המייל יכול לענות על השאלה.
Hi. Very, very nice talk. Very interesting stuff. So I have a friend who I worked with, and he's now at UCL, and we worked on her with a mouse cast. There's basically Bayesian derivations and modeling, and we came up with like some math for what we call reevaluation. So, we're basically what we're interested in is how new information or new observation causes reevaluation of the recent past. And it was for modeling behavioral tasks on mice, and it's very simple. Just two stimuli, a beep and a pure silence. And now that's kind of tied because the mice are not as smart as people. So who knows if they're reevaluating, but yeah, he wants to continue doing work on that project, and I really like the model, the math that we did. So if you're interested in I guess in the framing of reactivation, I would kind of ask like, are reactivated events reconsolidated given the current event boundary?
Yes, I think that is definitely true. So this notion of re-encoding.
Yeah.
So here's another project that we're running. Let's say you've listened to my talk, and then I tell you, well, actually, I've never done this. This is like
it's all Inkscape.
Yeah. That would mean you would need to integrate that information, previous information, because that would change things.
Yeah.
So
a plot twist in the movie.
Exactly. This is what we're doing right now. We have data of people watching it.
Okay. So if you're interested, I would love to talk to my friend who's at UCL, see if we could put together the write-up of the math that we did. And if you're interested in the it's basically saying that this reevaluation occurs from the Bayesian perspective because of a joint likelihood.
Yes.
And in it, there's the equivalent.
What do you mean by the joint likelihood? Basically, it's just a term, the mathematical term.
Yeah, P of X given A and B.
Okay.
So the probability of an observation given, the probability of the scene.
Yeah, given previous.
Yeah, and it's given the latent structure.
The latent structure. Okay.
Yeah, so the way I would.
So it's more like how I think about a model rather than the actual experience.
So the observations, the experience, and the latents are the model.
Right.
So you're basically saying, oh, I'm going to dramatically change my model given this.
Exactly. The transition structure will be different, and
So what we actually do is instead of focusing on when you say transition structure, are you talking about the transition like a transition matrix? So what we are focusing on is actually the likelihood of the likelihood function, which is the probability of observations.
Yes.
And that results in, so the transition structure results in like exponentially decaying similarity to the past, or to because it's every single time point. It's the joint likelihood can result in this renormalization where it's not exponentially decaying. So a far distant past now becomes more relevant.
Yes.
So this is the stuff that we focused on. We have like a model of it where it's both in terms of Bayesian inference, which has its own issues, and then there's also a memory-based estimation.
בסדר, הרב ג'ון.
Yeah.
אני רוצה להגיד, אני סטודנט של יובל הרב. איציק, דיברתי לך פעם הזה.
מה נשמע?
אז בסדר גמור. וכרגע אנחנו עושים אנליזה של הנרטיבס, אבל של הטקסט עצמו.
איך הולך עם זה?
עם איפה מראה קשוח, כי רצינו להראות משהו כזה מאוד שפטני.
איפה מראה קשוח?
נכון.
איפה מראה זה מעצבן?
אבל אני כזה לקחתי את יובל לקטע קצת יותר בהביורל כרגע, מתמקד בזה, זה הולך יותר טוב, ולא לדחוף בדרך כלל מה לעשות.
מגניב, אני אשמח לעזור.
אוקיי, תודה. כבר עזרת, אבל כן.
בשמחה, בשמחה ובהצלחה. אוי! אתה רואה את יובל?
כן, אני רואה את יובל. הוא רוצה להגיע היום, אבל הוא לא...
אני יודעת, עמוד.
אבל ביום ראשון אני רואה אותו.
אוי! זה קצת נשמע פחד.
בסדר גמור. שתדעו לקרוא את זה.
זה בסדר, תודה רבה.
זה היה ממש מעניין.
תודה,
זה היה מעבדה חדשה.
בעיקר שרדתי, זה מה שחשוב.
So yeah, I won't take too much more of your time. So yeah, if you're interested, we can send you a write-up. So my personal theory is that stuff's going to be seen in the OFC.
I completely agree. So I think about the OFC. I see it in all my analysis that survives correction.
For multiple persons?
Multiple comparisons. So I'm running my analysis across the entire brain. So I have to correct for all these voxels. So I see it. It's there. It just never survives.
Do you have an intuition why? Is it because the signal's weaker?
I think so. It's weaker in comparison to other brain areas that show higher reactivation. That's the problem.
Yeah, it's a lot more so. I mean, it makes sense. It's a lot.
Yeah, but I totally think it's
yeah. Okay, so I'll send you an email.
Yeah, sure. I'm Avital Hahamy. Hahamy.
We have the email about the talk.
Amazing.
Thank you so much.
Thank you for reaching out.
Thank you. Bye.
Bye bye.
אני אעביר לו את האות.
היי, אני אוריאן.
שלום.
רציתי להגיד לך שהיה ממש מעניין
איזה נחמד מצדך, תודה. תודה. אני באתי לפה ברגשות מאוד מעורבים.
כן, למה?
כי יש מאוד כמה מחלקה הזאת מפחידה.
בקטע כזה.
בקטע כזה, כן. וגם את יודעת, גם את יודעת, מישהו שעושה קוגניטיב כזה. אז זה נורא נחמד מצדך.
הייתי אומרת שזה קוגניטיב פלאט.
תראי. בהשוואה למה?
כן.
אז נורא נחמד מצדך, תודה.
בכיף.
כן, וגם לא הרגעתי שהם רואים עליי באופן מיוחד.
לא, נראה לי שהתגובות היו חיוביות. כן.
באיזה מעבדת?
האמת היא שאני לא מהמחלקה הזו בכלל. כן. אני בכלל כזה מתעסקת בפילוסופיה של Neuroscience. כן. במכון כהן באוניברסיטת תל אביב. אני פשוט גרה בירושלים, אז אני כזה באה למה שנראה לי מעניין.
כבוד, שעברתי קצת.
כן. לא, אבל אני ממש מתעסקת כאילו אני מנסה להבין דמיון. כן.
דמיון זה דבר מאוד מאוד מעניין.
כן.
אחת העבודות הקודמות שלי השוותה בין ראייה, דמיון ויזואלי והזיות. אז אולי זה יעניין אותה, וכן, זו עבודה שעשיתי עליהם סינדרום שנקרא סינדרום צ'ארלס בונה. זה אנשים שפעם ראו ואיבדו את הראייה שלהם בגיל מאוחר, ומאז הם חווים הזיות של ראייה. שמתי אותם בנט, אמרתי להם לתאר מה הם רואים, שראו שזה לא ראייה אמיתית. ואז יצרתי סרטים שהם סימולציה של הזיות שלהם, והראיתי אותם מבטאים שיכולים לראות, ונתתי לעיוורים אחרים שלא חווים הזיות לדמיין. וגיליתי שבעצם שלושת סוגי הראייה האלה מפעילים את אותם אזורי הראייה במוח. יש עוד חוקרת ב-UCL שחוקרת ספציפית דמיון, קוראים לה נדינה שם...
כן, זה ג'יסקה.
כן. והיא הקטע שלה זה דמיון. היא כאילו, היא מנסה להוכיח שדמיון הוא לא שונה מראייה.
כן.
זה לא תודה, זה יכול להיות פתאום.
כן. זהו, זה כאילו יש את המחנה הזה ויש את המחנה שאומרים שדמיון זה לא שונה מזיכרון.
נכון.
ואז כאילו דמיון הוא פשוט כמו כל דבר שאת כבר עושה.
כן, אני, לפחות לפי הדאטה שלי, זה נראה שדמיון הוא שונה משהו. אני מניחה שיש וריאביליות גם בין אנשים, עד כמה הדמיון שלך הוא חי. אומרים את זה? אומרים חי?
כן.
כן? אוקיי.
אפשר להגיד.
לא, בטח יש מילה יותר טוב.
נכון.
לא יודעת. בכל זאת מגניב.
כן.
אני שמחה שעניינתי.
כן, לגמרי. וגם מזל טוב על המעבדה החדשה. שיהיה מלא בהצלחה.
תודה רבה, ממש נחמד מצדך. תודה רבה, וגם לך. ביי. שלום. הוא גם אמר לי, רק נראה בשביל כן. אז הוא גם אמר לי, אני צריכה לצאת בערך ב3:30. וואו. סליחה, אני בשעון לונדון. אמרתי, וואו, אני מפחדת את זה עוד שעה.
לא. כדי לעשות את זה.